we going Today we're going to see drug target identification using systems biology approach. Let me switch on. Mm, Point, point, right. Okay. So first we need to understand what systems biology means and what's the definition that's given as per NIH. So NIH defines systems biology as an approach to understand the larger picture, be it at the organism level or tissue level or cell level, by putting multiple pieces together. It is completely different from the reductionist biology, which is what we have been doing and we have been taught for the many decades now. So the first thing that comes to mind would be like, what is reductionist biology? Reductionist biology is nothing but the way we are studying one entity at a time. So biological systems are complex. Everyone understands that. So to reduce the complexity, what people for many decades they ended up doing is that look at only one component at a time. So for example, if you are going to study about DNA replication, you'll study only the DNA replication machinery and not look at the other components that are influencing the DNA replication machinery. The assumption in that particular case would be that you are um, looking, you are assuming that the other entities have very little influence. So if you take any biochemistry pathway and biochemical analysis of pathways, you will see that everything is much simpler and easy to understand based on the reductionist approach that has been taken in like amino acid metabolism, carbohydrate metabolism, purine metabolism, pyrimidine metabolism, and so on. So this is the way that for many decades, as I said, reductionism was a method that was used in even in physics, chemistry, to look at complex things, understand it, and then try to replicate the mechanism in a particular way. There are a lot of advantages of reductionist biology. You will to understand the mechanistic view, you'll be able to understand the atomistic view of certain entities. However, you will not be able to look at the big picture because there are a lot of variables for which we do not know the answers about. So systems biology is looking at all the entities together in a single view. So this is completely different from a reductionist biology approach. And what you start with is you start with a model, and this is where computational modeling helps. And then you add cell biology components, then you add proteomics, then you genomics, and then some immunology, and then now you have the entire systems approach. Now, this is very similar to saying that if you have to look at a particular recipe, look at the ingredients separately, mix all the ingredients, and then now you have the recipe done. That's not how it works. So there was a lot of trial and error that happened during the initial phases of biological research, where researchers tried to understand how a particular bacteria moves towards a particular signal, like for example, chemotaxis, which is a signal transduction pathway in bacteria, the simplest pathway that's present. And if you are going to look at that particular chemotaxis pathway, you will have many entities at least 10 to 15 different proteins and many different metabolites, many different reactions. And so to understand how the signal is transduced in the cell, you will probably end up doing a lot of trial and error with it. So many years of research has gone into characterizing physiological and biological biochemical mechanisms. And then the question that was there for many, many years was that does biology follow a lot of the deterministic laws and rules that are there in physics and chemistry? And as you might have seen with the progression of um, the biological natural science history, you will see that biology also follows the same laws of physics and chemistry, and it does have deterministic laws and rules. So, so biochemistry textbook, as I said, is a classic case where you have a particular way of reducing, um, let's say you want to talk about wine, then it is basically grape juice, which consists of carbonic acid and alcohol. But then that's exactly not what happens in wine. There are many other small ingredients which are negligible amounts, but they do form a um, influence the taste, influence the characteristics, and different other properties. So this reductionism, let's go a little bit into detail as to why it was an easy way to understand things. You can think about reductionism is something very similar to a strategy that was very helpful in understanding a lot of science behind a lot of entities. And this can be traced back to Julius Caesar's divide and conquer, but it has its own limitations. Like, for example, as I said, if you want to understand an entity like cake, cake consists of flour, milk, eggs, butter, sugar, and candles. But a birthday cake is more than these ingredients. And if you put all these things together, you don't get a cake. You have not just the methodological part of preparing a cake, but also a lot of other variables that you don't consider when you're mixing flour with milk as it is. So most of the concepts in reductionist approach are fragmented, and the design principle is very, very less. So biology being non-linear in nature, there is a lot of ambiguity, and there is a lot of ways in which you can understand things. For example, you can have an experimentalist and a theoretician looking at the same thing, but in two different ways. An experimentalist will observe things and that cannot be explained. So they may say, okay, that is a reaction happening, but even the reaction is happening at a particular pH, um, we don't see the visible outcome. So an experimentalist will be able to observe a lot of things, but what is causing that particular reaction, they may not be able to explain. However, theoreticians can explain things, but they cannot be observed. For example, a theoretician will say that on the moon's surface, there is this reaction that can happen. But does it really happen in on the moon's surface? We don't know. But theoretically, you can explain those things. So what is the alternate here? The alternate is the reconstructionism, which is what in the 1970s and 80s, and one of the main influential person who talked about this and also helped um, in many ways the developmental biology circle is Sidney Brenner. So you have an emerging paradigm where, in addition to traditional experimentation, that is, you take the isolated parts, you put them together, but then you also monitor the interactions with the environment that goes with all this. Okay. It needs well-controlled in vitro experiments, of course. So whatever has been identified in the past you do need them to come up with models, you do need them to come up with explanations, but at the same time, you need mechanistic rate laws too. So you need three things. One is a means of simplifying the problem, a convenient terminology, and a convenient mathematical representation for large systems. So, for example, this is I took it from a book which you can refer in your free time. It's by the author called Ida Clip. And here you have a biological system which is a bunch of E. coli cells. They are producing thousands of different proteins at different points of time. They are all from the same parent cell, but however, the daughter cells within each daughter cell, the concentration of the proteins will be different. However, for the sake of simplicity, we consider each cell to be identical to other and assume that all the proteins are present with a particular ratio with respect to others. So you come up with a mental model. The mental model is basically a schematic of the same. So here there are two proteins, protein A and protein B, or protein X and protein Y shown in red and blue, and the different concentrations of protein X and Y shown. Now, protein X activates the formation of protein Y, and when you start describing the events that regulate one with respect to other, you have a scheme. And in the case of systems, an arrow indicates activation, and an arrow with a horizontal or a vertical line will indicate inhibition or repression. This is basic biochemistry. So we use the same nomenclature. Now, with particular concentration of X and particular concentration of Y, you cannot just start with saying X influences Y. You have different setups, like when X is not present and X is present, when X is present and X is not present, and vice versa for Y. So you get different constants, rate constants of X being produced and Y being produced. And that leads to a dynamical model. When we talk about the word dynamic, we incorporate time. So dx by dt, dy by dt, which means the rate of production of X with respect to time and rate of production of Y with respect to time. So you can come up with equations, and then when you simulate the same setup, you get a quantitative result. Quantitative here being that you can measure how much X is being produced at a particular point of time, how much Y is produced at a particular point of time. So from a biological system, we used reductionism, but then we reconstructed the interaction of X and Y with respect to time. So this is a very simplistic way to explain what we call as abstraction. Abstraction means that you are converting complex scenarios or complex entities in very simple steps. So when you have that kind of a model developed, you have a lot of reasons and a lot of advantages to develop such kind of models. First is that it drives conceptual clarity. So if we go back, if I have to study all the 4,000 proteins at the same time, I can do that. But studying two proteins at one time and then looking at the quantitative dynamics between them will lead to more questions, and then I can slowly add more proteins to it until I reach all the four proteins and the dynamics of all the 4,000 proteins. So, and also it highlights gaps in knowledge. For example, if I have to go back to the same and then I say, okay, I know the rate constants of X and Y. So I can easily come up with this model. But let us say there are proteins for which I don't know the rate constant because those kind of assays have not been completed or not been done in a wet lab. Then I can identify, okay, I need that particular data using which the model can be constructed. So it also provides independence of the modeled object, meaning that if I have to look at a very modular approach of certain components and not all other components, I can do that. Now, time is an arbitrary unit. It can be stretched, it can be compressed, and because when we look at time in a cell, the time taken, it is not in the same time. The time scales are different. DNA replication happens at a different time scale in uh, seconds, whereas chemical reactions, like for example, phosphorylation, happens at a different time scale, which is in nanoseconds. So when we are looking at one single model and we are looking at different components, each component can have a different time scale, which means that I can run the simulation fast or slow, so I can reduce the time or stretch the time. And similarly, I can do it with space. Space being is either I'm looking at a particular part of the cell or the entire cell. I can limit the space. And of course, there are other advantages, just like how you do with computational modeling. There are a lot of ways you can help experiments, you can generalize, you can test a lot of different hypotheses using the approaches. So once you once you are trying to develop a model, the most important concept that comes up is scope. Now, scope of a model is the first question anyone will ask you when you're trying to when you're trying to say that, okay, I have a computational model of, let us say, chemotaxis in bacteria. Then the question is, what is the scope of the model? Scope is nothing but the properties of the system being studied. Can you describe those properties in quantitative terms? When you describe them, that becomes the scope. That means whatever property I'm unable to study, it's out of scope for that particular model. So you construct models that disregarded properties and do not compromise the basic results. So what are the elements in the model? What are the number of species? What are the equations? We'll come to these equations later. What are the constraints? Okay. And what type of model it is? That also determines the scope. Is it a static model? Is it a dynamic model? It is a deterministic model. It is a stochastic model. What are the different variables used? What are the parameters? What are the constants used? All these things, when you answer, you automatically are trying to define the scope of the model. So I hope most of you might know what deterministic and stochastic is. If not, this deterministic means that if you are running the model every time, it will give you the same answer. That is called deterministic. Stochastic, when you run the model, depending on the parameters, every time you run, it will give you a slightly different answer. So that is called stochastic. So if it gives the same result again and again, that's deterministic. Stochastic, there will be variation in the outcome. And there are both pros and cons of having a deterministic model or a stochastic model. So, for example, this is a system. This is starting with glucose, and glucose EX here indicates that it is external, and glucose IN indicates internal. So glucose from outside comes inside. There's a rate at which the glucose can come inside the cell, or it's transported inside the cell, and so you have a reaction rate. Once the glucose is inside the cell, it will you can convert it into, if the initial part of the glycolysis pathway, it gets phosphorylated, so it becomes glucose 6 phosphate, so that is S2, and then something is being produced from glucose 6 phosphate, that is S3. It is transported outside, or it goes through a different direction, or the glucose 6 phosphate is converted into a fructose 1,6 biphosphate, S4. Okay, so the legend is given here. And then in this particular step, you have ATP being used. So A3 is ATP and A2 is ADP. So ATP is used up, so the phosphate is taken up by the substrate. Now, what you see in the dotted lines is the limit of the system. Let us say the rate at which the glucose enters the system is constant. The rate at which glucose 6 phosphate is taken out of the system is constant. 1 second. And the rate at which fructose on 6 biphosphate is being produced is also in a different way, a kind of a constant, or you're not worried about it. Then these three entities, we call them as species in systems biology, S0, S3, and S4, are outside the limits of the system. Whereas here, the number of ATP being used is outside the system. In this particular case, the limit is everything is part of the system. Okay, so you have the same reaction setup, same set of reactions, or same set of pathway, but the dotted lines indicate what is the limit of the system. Now, why are we talking about limits? Because for any system in systems biology, it has to reach an equilibrium. That is in biochemistry, we call them as steady state. So steady state, only when steady state is achieved, you can understand the system and you can do more analysis and you can do other type of systems biology approaches to it. How do we decide where is the limit? That depends on which particular definition of the system, that is a scope, is giving a steady state entity. So if we have to simulate this particular, the first system, you will see the lines that indicate with respect to time, and very few components are reaching steady state. In this particular one, where you do not have a limit, everything is part of the system, you have certain things going near zero and they don't reach a steady state. Whereas in this particular, you have a steady state achieved and you have certain things remaining in constant with a after a particular point of time. So when we take a basic metabolic network, you have a metabolite that takes part in a reaction, gives another metabolite that takes part in another reaction and gives a metabolite, and so on. So you can think of this as a cascade or a series of reactions. So when you have identified the metabolic pathway or the system that you want to study, you first have to set the limit of the system. And as I said, you set the limit and then you have to check for steady state. And if steady state is not achieved, then the limit has to be changed. Then you have to balance the equations. Now, balancing the equation, we know this from basic chemistry, even in your school. We would have done like you have to balance the left hand with the right hand and so on. And then you have to add the empirical values of the reaction, which is the assignment of kinetics. So this we use it from the reductionist data that we have, reductionist approach data that has been studied. We know all this data for many different reactions. So as I said, in reconstructionism or in systems biology, even though it is not reductionist approach, we take the reductionist approach data to understand or build the system. Okay. Okay. Now let us say we are taking this particular system and we are we are looking at the different entities. So as you can see, we are looking at organic molecules from glucose to fructose 1,6 biphosphate. And here in this particular instance, since these are organic molecules and we are looking at living systems, we ignore the phosphate and we only see the balancing of carbon. Now, carbon balancing is the one major view with which we describe a system, because that is the way we can understand the balancing of the equation. Does it mean that do we ignore phosphate, or does the system ignore phosphate? It doesn't, but we would like to focus only on carbon chemistry. And the reason is that it most of the reactions, most of the equations, most of the empirical data that we have is carbon centric. We do not have many other metabolites centric like phosphate or potassium or iron or sulfur. All those things are part of the network, but we do not have many different empirical data to describe the system in detail. So once we have this particular setup, we first write the ODE. ODE stands for ordinary differential equations. Ordinary differential equations are very easy. There are many different equations that you can come up with for systems approach, but ODEs, or the ordinary differential equations, are the easiest to explain a system with respect to time. There are partial differential equations, there are stochastic, there are quadratic equations. You can come up with many different other complex equations, but ODEs are usually what is used for biochemical pathways, which is what we are concerned about, because most of the time for drug designing, we are looking at whether a particular target structure plays a role in the cell, as and up to what level. So then in that case, we need to create ODEs. Then once we have the ordinary differential equations, we have to balance those equations. And to balance the equation, you create something called as stoichiometric matrix. Again, those who have studied chemistry in the school, you would have balanced equations using a stoichiometric coefficients. Okay, so stoichiometric coefficients are nothing but the proportion of the entities. Here you have one entity of S1, one entity of S2, giving rise to two entities of a product. So the stoichiometric coefficient of S1 is 1, and stoichiometric coefficient of S2 is minus also 1, and the stoichiometric coefficient of the product is 2. However, if we look at the forward reaction, S1 plus S2 gives rise to 2P. So S1 is used up in the reaction, S2 is used up in the reaction, and the product is produced. So whatever gets used up, you give a negative sign. So the stoichiometric coefficient gets a sign -1. For S2 also -1. For P, the stoichiometric coefficient will be +2. This is in the forward reaction. In the backward reaction, it will be -2 +1 +1. Or if you are going to look at stoichiometric coefficient, you can have it as -half, and 1. Okay. All these are valid stoichiometric coefficients, and it depends on which one you're taking into consideration for building the model. Usually, we like integers, we don't like fractions, and usually the convention is to use the forward reaction even if it is a reversible reaction in the cell. So we usually go with 1, minus 1, minus 1, plus 2. Okay. Now, the ODE is ds1 by dt. The rate of S1 being used up by dt is given by minus V, because S1 is being used and there is one reaction it is taking part, which is given with the convention V, or you can say to be specific V1. So the ds1 by dt is minus V1. Same ds2 by dt will also be minus V1, but dp by dt will be 2V or 2V1 subscript 1. Okay. So this is the ordinary differential equation for this particular reaction. So for a metabolic network, when you expand it to m number of substrates with r number of reactions, you want to balance the equation. You will describe all the reactions with respect to the substrate and or the substances and the number of reactions. So where n, I, j are the, sorry, there is some. One second. Yeah. So, where n, I, j are the stoichiometric coefficients for the ith metabolite and the jth reaction, where I goes from 1 to m. So the summation of all those stoichiometric coefficients will give rise to your equations. Now, external metabolites are not defined in the model. One second, I have to mute myself 1 second. Yeah, let me share the screen again. I know I am not speaking at the moment. That's why I'm not audible. Right. I had a bad cough. And so, yeah, so external metabolites that are not defined are kept constant, and thereby you are keeping them out of the equation. So you assume that the external metabolites don't play a role and do not have any impact in the system. So the stoichiometric coefficient, Nij, you assign it to the number of compounds that are there in the system that you are taking up into study, that are taking part in the number of reactions Vj. J goes from 1 to n, and you are making the stoichiometric matrix uppercase n. So this is the name notation that's given for the stoichiometric matrix. So what you see here is the stoichiometric matrix. On the rows, you have the substrate or the metabolites. So S1, S2 up to Sm. And V1, V2, the columns in the matrix are the reactions. So N11 is nothing but the stoichiometric coefficient of S1 taking part in V1, S1 taking part in V2, that is the second reaction, it will be N12. So you will have a set of coefficients that you will fill it up in the matrix. Those reactions for which S1, S2 don't take part, you will put 0. Wherever you have reactions for which the substrate or the substances take part in, it's only in those part of the matrix that you will fill. So what does this stoichiometric matrix tells us? So this matrix tells us that the reactants and products take part in a specific reaction in the model, and the stoichiometric value tells us what is happening with the reactant and the product. What specific species the reaction is part of, whether that species is a reactant or a product, we know that by the sign, plus or minus. So it is a M by R matrix where M equals total number of species, R equals the total number of reactions. So by convention, we give the species in the row and we keep the reactants or the reactions in the columns. Now, while we looked at reactions only in the forward direction, we do not have information whether they are reversible or irreversible. Although you can say, okay, because it is in the forward direction, every reaction is irreversible. That's not necessarily true. And for irreversible or for reversible reactions, as I said, we only take the forward direction. That's by convention. There's not necessary that you have to do that. But if you have to take the backward reaction, you have to add another row to indicate the backward or the reverse reaction. Because, sorry, indicate another column to indicate the backward reaction. So, as I said, the direction of the reaction is assigned positive when you go from left to right or negative from right to left. So, the mathematical description of any metabolic model can be described in terms of a mathematical expression that consists of vectors, and there are three vectors which are required to describe this particular or a particular system. The first vector consists of the concentrations of the substrate. The second vector is a vector of reaction rates, and the third vector is of the parameters, and then you need the stoichiometric matrix. Now, one using all these three vectors, you can describe any system in this particular manner. For example, S1 plus S2 plus S3 takes part in one reaction V1 and gives rise to S4 and 2S5. Now I'm going to make this interactive. What will be the stoichiometric coefficient of S1? You'll have to unmute and say it or put it in the chat box. This particular slide is very interactive. So yeah, Alekia says 1. How many of you agree with Alekia? Okay. Participants, you have to unmute and say, be interactive in this particular session. There are 31 participants.
So can you repeat the question, what you asked?
What is the coefficient of S1?
1, sir.
How many of you? Anyone else? Okay. So Ankur and Alekia are correct. It is 1. Okay. So what I'm going to do is, we have 31 participants or 30 participants or 28. excluding me and Radhakishan ji, the more I hear from you, I'll tell the organizers to give marks to you guys. Okay. If you are quiet, then you get 0. Okay. Now, what is the coefficient, stoichiometric coefficient for S2?
1, sir.
1. What is the stoichiometric coefficient for S3?
1.
1.
One.
What is the
One.
stoichiometric
One.
coefficient for S4?
1.
1.
One.
And what is the
One.
stoichiometric quotient for S5?
2.
Two,
Two.
sir.
2. Okay.
Two.
What will be now looking at the forward reaction? What will be the sign for the stoichiometric quotient for S1?
Minus.
So minus 1. Okay.
Yes, sir, minus 1.
For S2?
Minus 1.
For S3?
Minus
Minus 1.
minus one.
Minus
Plus 1.
S4?
Plus 1.
plus one.
Plus 2.
S5? Plus
Plus
2.
2.
Plus
Plus
two.
Okay. Now, how many reactions are this species taking part in? How many reactions are there?
One.
1. Good.
1.
Indranil said good. Okay, 1. 1 reaction. So if we put reaction in the column, how many columns will be there in your stoichiometric matrix?
3.
Three, sir. Three.
No. How many reactions are there? 1. So
1.
reactions are
reaction.
in column. So how many columns will be there? 1. Okay.
One.
One.
One column.
So, yeah. And how many rows will be there? 5 rows. So now you
Five.
see in the
Five.
screen, this will be your stoichiometric matrix. -1, -1, -1, 1, 2. Why? Because this first row is for S1, second row is for S2, third row is for S3, fourth row is for S4, and fifth row is for S5. So the
Yeah.
signage and the coefficient tells us the stoichiometric matrix. N equals -1, -1, -1, 1, and 2. Okay. Now, this is minus and this is plus because we are looking at the forward reaction. If it was reverse, what will be the stoichiometric matrix?
Minus 2.
No,
Minus
the
1. Plus 1.
row will not change. Row will not change.
Okay.
1, 1, 1, minus
1.
1, minus 2.
Exactly. Gajal Gupta said it right. 1, 1, 1, minus 1, minus 2.
Minus 2.
Okay.
Okay.
Now, how many substrates are here? How many substances are here?
Four.
4.
4.
Four.
4. How many reactions?
Five.
5.
5.
What will
Five.
be your matrix?
1.
What will be your matrix? How many rows
No,
and how
no.
many columns?
Sir, column one. No.
Column एक आएगा कितने?
Column will
Every
be 4
reaction is a
and
column.
row will be 1.
1 by
Every
5.
1
5 columns क्योंकि every reaction is a column.
column. 5
Okay.
columns.
5
and
columns
4
and how
rows.
many rows?
4 rows.
4 rows. Okay.
Row.
Now S1 is taking part in how many reactions? I'll start with this and then you can work out the rest. S1 is taking part in V1 and
And
V2.
V2.
So S1 coefficient, what is the coefficient? 1. Okay.
1.
In V1, is it produced or is it being used?
It is
In V1.
produced, sir.
It is
Produced.
produced.
Just
When
for
it's produced, what is the sign we give?
Plus 1.
Plus
Plus
1.
one.
Okay. So in the first column, you will put plus 1. In the second column, which is for V2, S1 is used.
Used.
So
Minus.
S1 will get a coefficient of
Minus
Minus 1.
minus 1.
Minus 1.
minus 1.
What about S2?
Plus one.
Plus 1.
Plus 1.
And
Minus 1.
And son.
then
S2 will
S3
be
Plus
minus
1.
1 and S3 plus 1.
is used in it's produced in V3 and used in V4. So you will
Plus
have
1.
the coefficient 1 with the proper signage, right? So you said 5 columns, 4 rows.
Hmm.
Coefficient matrix will look like. This is for S1. Now S1, it's taking part only in V1, V3, V4, V5. So it gets 0. Okay. Now automatically you will see that this is not a symmetrical matrix. Okay, so the top triangle does not equal the lower triangle, and there is no diagonal because your rows and columns are for different entities. Okay, a little bit tricky. First, tell me how many will be. Count the number of V1, V2, V3.
Three.
Three
Three.
columns.
Three.
How many species are there?
Uh, 3. So
Three.
3.
Children, yes,
Three.
three. S1, S2, S3. But S1 is taking part in how many reactions?
Two reactions.
Three
Three.
Reaction.
reactions.
Two.
V1, V2, V3.
With three.
S1 has a coefficient of 1. In V1, it will get the signage
+1.
plus
Plus
1.
one.
In V2,
-1.
Minus one.
V3,
Minus
Minus
one.
one.
Minus one.
now in S2 is taking part in how many reactions?
Two reactions.
Two
Reactions.
reactions. Okay, so in V1, the first column S2 will be 0. In V2, it is used or produced?
Used.
Used
Plus
up.
Used.
use.
Minus
Minus 1.
Minus one. Minus
Okay,
one.
minus 1. In V3,
+1.
plus
Plus
1.
one.
The other way around for S3. Okay, so your stoichiometric matrix will be like this. Okay, S1 is produced in V1, used in V2, V3. S2 is not taking part in V1, used in V2, produced in V3, and the other way around for S3. Okay, so this is how you construct the stoichiometric matrix. When you construct the stoichiometric matrix, you automatically balance the equations. Okay, now can everyone do this? I'll give you 2 minutes time. Okay, take a pen and paper and first let us we'll do this along with you. First, tell me how many reactions are there?
6 reactions.
six
Sure,
Reaction.
sure.
6 reactions.
Seven.
7.
Seven reactions. Okay. How many substrates are there or how many metabolites are there? 1, 2, 3, 4, 5, 6. You will not count this as 7 and 8 because ATP is occurring only once. Okay. So 6. What about this? What is PI? That is phosphate. Okay.
inorganic phosphate.
We are ignoring phosphate, remember? So we are looking at only carbon chemistry. We are not looking at other metabolites. So we'll ignore those. Okay. So does phosphate get a row in the matrix?
No, sir.
No,
No,
it will not get a matrix. Okay. Now for the first column, what will be the values?
+1.
+1.
Plus one.
And then
Minus one.
Minus one.
-1.
no. Okay, you're doing it row wise. Okay. So +1, -1. Glucose is taking part in V1, V2. Okay. And then after that, glucose is not taking part in any of this. So it's 0.
0.
No.
Glucose 6
Zero.
phosphate is taking part in V2 and V3. So you have 1 and -1. Now look at these two rows. Okay. This row is for ATP. This row is for ADP. In V2, ATP is used up.
-1.
So
Used.
-1. V1, V2, V3, V4. What is happening in V4? ATP is used up. So it is giving -1.
Minus one.
But in V7, ADP is converted to ATP. So ATP is produced. So you get a +1.
+1.
Okay. So this is a stoichiometric matrix for the same setup. You understood now how to construct a stoichiometric matrix?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. Now, once you have that, as I said, we have to construct the ordinary differential equations. So take the same reaction, S1 plus S2 plus S3 gives to S4 and 2S5. S1, S2, S3 is being used up. So ds1 by dt, ds2 by dt, ds3 by dt is minus V1. There's only one reaction, V1. ds4 by dt is plus V1. Now, the coefficient, what is the coefficient here? It's there, 1. Okay, in both these cases, -1 and +1 is present. But since the convention is until unless it's a integer of 2 or 3, we don't put 1 specifically. You don't put -1 V1. It makes a lot of sense to just put minus V1 or plus V1. For ds5 by dt, it's 2V1. Okay, so now this is the ordinary differential equation. This is for the same set of reactions. ds1 by dt, since it's taking part in V1 and V2, V1 it is produced, V2 is used up, so V1 minus V2. ds2 by dt, V2 minus V3. ds3 by dt, V3 minus V4, and so on. Okay. In this particular case, ds1 by dt is V1 minus V2 minus V3. So one ordinary differential equation can be constructed for every species or every substance that is present in the reaction. Now tell me what will be ds2 by dt in this reaction?
DS
ds2
Minus
is
minus
V3
V2.
minus V2.
minus?
V3 minus V2.
Why minus v3?
minus V2 only.
No, but it's taking part in V2 and V3. So V2, V3 has to be present. What will be the signage?
Minus V2 plus
Minus
V3.
plus V3.
Or V3 minus V2.
V2.
Okay. What will be for a ds3 by dt?
Sir, plus V2 minus V3.
Plus
Okay.
V2 minus V3.
Okay. Understood? So these are
Yes.
the ordinary differential equation. Now, why do we have to construct this ordinary differential equation? I already gave a little bit background. Because we have to, once we have constructed the model, we have to see at what time point does it reach steady state. Only when the equations can be solved as differential equations. If you solve the differential equation, you will know that you have minima, maxima for every differential equation, which is what you would have studied in your calculus classes. And so you can actually find out where it is maximum and where it is minimum. And you can find out at what point they reach a steady state. So tell me what will be the ODE for D glucose by dt?
Plus V1 minus V2.
Glucose is used. V1 minus V2. Okay. Similarly,
Yes.
glucose 6-phosphate, V2 minus
Minus
V3.
V3.
Okay. Now you can see for this one, DATP and DATP by dt, it is the signage is different, but otherwise it's the same V2, V4, and V7. Okay. Now, what you see here on this left side of the screen is for every reaction, you'll have a rate constant. And these values, K1, K2, K3 up to K7, those values you get from experimental data. Like for example, someone would have done the experiment of rate of glucose being utilized when you phosphorylate. What happens or what is the value, empirical value? All these data, you get it from experimental results. So we use that along with these to simulate the reaction. Now let us say K1, K2, K3. Now what is K1, K2, K3? Depending on the reaction V1, V2 up to V7, each reaction has a rate constant. if we consider K1 as 0.25, K2 and K6 as 1, K7 as 2.5. Let us say you start with these rate constants, and then you have a steady state concentration, or you want to find out at what point they reach a steady state. The AU here represents arbitrary unit. This doesn't have a unit. It's not seconds or minutes. It's just that at 10, 20, 30, 40, so on. We go from a very low concentration, and slowly you can see at almost at about 20 unit time, almost everything starts coming into a steady state kinetics. At this time, we can see that the reactions we wrote for this system, which is only these 7 equations and these many substances, you can find out the rate at which glucose is being produced or used up, glucose 6-phosphate being produced, and so on. Now, this is with respect to concentration of the organic molecules. Okay. Now, if we have to look at in terms of ATP, how many ATP is being used from 0 to 1? You can see depending on which particular reaction, it will be different. Now, this is the same setup, but since we are looking at in terms of ATP in the x-axis and not time, the properties of the system is the same, but the representation of the data is different. But you can immediately see that ATP is being produced or used up, ADP is being produced or used up, depending on which particular metabolite is produced or used up. So what you get at the end is a steady state concentration. How much of the ST represents here steady state? What is the steady state concentration of this particular network or this particular metabolic pathway at a particular point of time? Now, you generally, in this particular previous slide, we started all with zeros. Okay, 0 glucose, zero glucose 6-phosphate, and so on. They accumulate until the production and degradation are balanced. Now, when the rate at which something is being produced equals the rate at which something is being degraded or removed, we call that as a steady state. That is the equilibration or equilibrium state. So the ATP rises and decreases at the same rate as ADP decreases and rises, because conservation of adenine nucleotides is assumed here. We assume that ATP and ADP, they have the same number in the reaction, because we are not considering phosphate as part of we are taking phosphate as part of the reaction, but we are not balancing phosphate. Okay, so you have to remember that certain inorganic molecules, we do not balance them, but we do take part or we do consider them as part of the reaction. Okay, so in this particular instance, the vector concentration vector will consist of these substances. The reaction rate vector will consist of V1 to V7. Number of reaction is 7, and the stoichiometric matrix is this. Okay. Now, here comes another concept called flux. Now, flux is something that is measured in a metabolic pathway or a metabolic network. When we talk about ordinary differential equations, something is being coming into the system and then something goes out of the system. So at a steady state, the rate at which something comes in and the rate at which something goes out is constant. So you will have different rates at which the entities come inside the system and go out of the system. The rate at which a metabolite is entering a particular network or a pathway is called as a flux. So the flux can be measured, and what you see here, when we plot for the same network, the darker the symbol is the sink, that is a negative flux. The lighter the color, the positive flux, which means that we can see for which particular reactions something is a sink, meaning that something being used up more than compared to others. So this in a 2D representation looks like this, but actually you can think of it as a valley and hills, and a flux goes through from the top of the hill to the valley. Now, in systems biology, we can look at metabolic pathway or metabolic reactions as networks. And when we look at it as networks, we can represent it in using a computer science concept called as graph theory. Anyone familiar with graph theory?
No, sir.
Okay.
No, sir.
So, how many
No,
of you have heard about Facebook?
Everyone, sir.
Everyone knows
Yes.
Facebook, right? So, what is the 6 degree of separation? Everyone, has anyone heard about it? 6 degrees of separation.
No, sir.
Okay,
No,
no,
sir.
so
sir.
No, sir.
degree of separation between me and take a very well-known celebrity. I see the name Lokesh here. Lokesh, who is your favorite celebrity?
Salman Khan.
Salman Khan. You know how many degrees of separation are between me and Salman Khan, or you and Salman Khan?
Yes, sir. Very large.
Only six. Okay, or not more than six. Okay, what is the 6 degrees of freedom? Oh, sorry, 6 degrees of separation. Everyone is connected to someone else. If you are a node, let us say in a network, you are the node, your relationship with that other person is the edge. So that is what we call the terms in graph theory, nodes and edges. So an entity is called a node, and the relationship between two entities is called an edge. So every node in a graph is connected with every other entity through a edge. Now, some nodes are connected more frequently, some nodes are not connected more frequently. So, for example, someone who is a public figure will have more number of edges, which means they know many people. Someone who is an introvert or who is always like not very outgoing or socially outgoing, they will have less number of edges because they know only so many people. Okay, so now let us say between you and Salman Khan. Okay, Lokesh. So Lokesh, which city you are in?
Udaipur, sir.
Udaipur. Okay. You might know someone in Jaipur.
Yes, sir.
Okay, so your degree of separation is one with that person in Jaipur. Okay.
Okay.
Does that person know any Bishnoi?
Vishnu, no, sir.
Someone from, like, okay, his classmate might be from the Bishnoi community. Okay, let us say. Okay, let's assume. Then that degree
Okay.
of separation is two. That Bishnoi community, do they know Salman Khan? Like, do they know?
Yes, sir.
They definitely would know because he shot a black buck, right?
Yes, sir.
So number of degree of separation might be three, or let us say even if there's one more person, it will be four. Okay. So if I let us say my degree of separation, let's try with some other example. I see the name Adnan here. Adnan, who is the celebrity? Your favorite celebrity?
Sir, Salman Khan.
Well, we've done
Actually, sir,
Salman Khan.
Adnan said that time also
Oh, okay.
on my
Then
behalf.
Lokesh. Okay, Lokesh.
Yes, sir.
I can't see the name and the voice, but Lokesh, who is your favorite celebrity?
Dr. Mohan Rao.
Who's that? I don't know him. Okay, anyway, let's say Mohan Rao. Okay. So you know someone and they know someone and they know Mohan Rao, or they know someone who knows Mohan Rao. Okay. Now this is what we call as 6 degrees of separation. Number of people on this earth is 8 billion, but everyone knows everyone else, or everyone is connected not beyond a 6 degrees of separation, and that's the whole concept of Facebook. If let us say, like let me give you an example from my side. The number of degrees of separation between me and Obama is uh, I'll keep charting it. You can count. So I know the vice chancellor knows the prime minister. Prime minister has met Obama. So the number of degree of separation is
4.
yeah. Okay.
Four.
So it's not just acquaintance. It's not just you know someone. They have interacted with them. Okay. So when we look at now coming back to systems biology topic. Now when we look at a network or a metabolic pathway, we can represent it as graphs. So in graphs, you have nodes and edges. Now, if you look at metabolites as nodes, glucose 6 phosphate, fructose 1 6 phosphate, all these green dots here are nodes, and the reactions they take part in, like for example, glucose and ATP, ADP take part in a reaction. So you connect an edge between these three nodes. So that you can construct a graph like that. You can construct with respect to metabolites. You can also construct a network using common reactions. Sorry, this is common reactions and this is common metabolite. Okay. So what we see here is in systems biology, a metabolic pathway or a metabolic system or anything can be understood in terms of a network. And because it is represented as a network, this is computer readable because you're listing the number of nodes and number of edges. You are talking about degree distribution. Like for example, ATP, ADP, you have many number of edges. So the degree distribution for ATP and ADP will be very high. Whereas for the GLYC here is only 1. The degree distribution is only 1 because there is only one edge connecting to it. You will have many different edges also present depending on what type of reaction you are looking at. So when we look at uh, systems biology, you can create ordinary differential equations, you can balance the equation, you can calculate the flux. Now, once you understand the flux, then there is another approach that is constructed or can be done called the flux balance analysis. So this we go to the next topic. Up to this point, anyone has any questions? Okay. Okay. So what is this flux balance analysis? The rate of turnover of molecules through a metabolic pathway. That is what we call as flux. And we are balancing this flux with the behavior of how much of the substrate is being converted into a product with unit time. So in a particular pathway, the regulation of flux is always happening. The regulation of flux means that it's vital for its activity under different conditions. Now, why do we need to study this? The reason we need to study this is that, and especially why we need to do flux balance analysis in a drug discovery. Let us say, and I'm going to give you an example. Uh, what are the between two cities? Let us say Delhi and Jaipur. The most frequent way people travel is by road. Okay, so many people go by the bus, or they take the car, or they go in a bike, they take the road. Let us say there is a huge traffic jam created, and that particular national highway is blocked. What will happen?
They go by train or by flight.
They'll go by train or flight. But let us say you are in a car, you can't take your car in a train or flight. What will you do?
We have to wait.
We can wait, sir.
You can't wait. Okay, assume you can't wait. Then what will you do?
We'll come back. We don't go. We stop.
What do you look for an alternate route to go to your destination?
We choose the another pathway.
That's what. So you will look at another state highway, or probably something that goes through one like a small road, takes a diversion, right? So if if there is a, let us say, a flux, that is the behavior of the metabolite going through a system, what happens when that particular flux is blocked? It redirects the flux through another network. Okay, and this is the whole reason why in systems biology, when we talk about a drug target, we think, okay, this is a particular target, this target is very useful. It can be, there can be a drug that can be designed and inhibits the performance of the particular protein, inhibits the protein. But does the cell die? The cell may not die, or the bacteria may not be killed. Why it doesn't get killed? Because that if you are blocking or you're preventing a particular protein's function, within the cell, there are other uh, associated networks that can be present, which will lead to an alternate pathway where the same function can be performed. Like for example, you if the glycolysis pathway is blocked, there will be a different way in which the same glycolysis pathway will be activated, or the pathway will still function, but not in the same efficiency. To give you another metabolic network example, in eukaryotes, you have isoforms. You have every protein exists in different isoforms. So when there is one isoform which is the dominant, let's say there is some mutation that happens and that protein is non-functional, then what happens? You have the other isoforms which compensate the loss of this particular protein, and the cells will still continue to survive, cell will continue its function. So in a drug design or in a target identification uh, uh, pipeline, you first have to look at the flux, or you have to look and see if there is any compensatory pathway that can be activated when this uh, particular target is inhibited or target is identified. And apart from drug design, we use flux balance analysis for many other things. Like, for example, there are a lot of marine bacteria which produce anti-tumor, anti-cancer drugs. These anti-cancer, anti-tumor drugs are very complex. You cannot synthesize them in the lab. Now, the bacteria produces these in nanogram quantities. That means you cannot commercially produce them in large quantities, um, and you need to redirect the resources of the cell to produce this in at least some microgram quantities. That means you have to redirect the flux from other processes and redirect it so that it produces this particular product or this particular anti-tumor drug in large quantities. Natural products, we call them as natural products. So there's a lot of work that has been done, and this area is called strain optimization. When you look at papers that talk about strain optimization, what are they optimizing? They're optimizing the flux that is directed towards one particular pathway into some other pathway. Like, for example, ethanol production or malonate pathway or some kind of secondary metabolites which are industrially important. Strain optimized pathways can produce the same metabolite in higher quantities. Okay, so to do that, you first need to do this flux balance analysis, which is a mathematical model that predicts quantitative values on importance of various reactions in a cell. And thereby you can test the hypothesis in an experimental setup. For example, there is most common one that we keep doing in flux balance analysis is we increase the biomass of a particular organism so that you can have the large quantity of a metabolite being extracted. So in experiment, how do you measure the flux? In experiment, you can use isotopes and you can find out using NMR or GCMS or other types of spectroscopy method, like by labeling the carbon with C13, you can find out how this particular flux is traveling. And you can find out, just like in a highway, you have national highway, you have state highway, you have minor and major highways. You can find out what are the major pathways in a cell where flux usually travels or is directed, and minor pathways where the flux can be redirected if the major pathways are blocked. So using this, you can find out the topology or the architecture of the network. Okay. Now, the central premise is that you can predict the metabolic behavior and you can do a selection of the productivity or efficiency of the metabolic networks. And that is what we do in flux balance analysis. We are not looking how the distribution is achieved. We are looking at which flux is distributed optimally in the cell. So this type of modeling is called constraint-based modeling. Now, constraint-based modeling is one where we are putting a lot of different constraints. What are the constraints? We are putting constraints in terms of mass, in terms of energy, in terms of the reactions. That is basically how the organism is behaving. The assumption is that the organism is at a steady state in whatever constraints is given. So, for example, if you have a bacteria, it is being exposed to a low water environment, that is in a drought environment. It will have some perturbations in the flux, but after some point of time, it will reach a steady state. There osmosis, diffusion, all those processes will happen at a very slow rate compared to the other processes. Similarly, you can think about a bacteria which is traveling towards a metabolite, which is a food, or traveling away from a metabolite, which is a toxin. So, one such constraint-based modeling is flux balance analysis, in which the stoichiometry of the network constrains the solution, which means that the solution is already there. You just have to locate where the solution is. So it cannot predict metabolite concentrations because it does not use kinetic parameters, and you can only do this analysis at a steady state, which means that to get a particular network to do a flux balance analysis, you have to construct the ODEs, you have to construct the stoichiometry matrix, and then reach a steady state, and then use that model, limit the model, define the scope of the model, and then you can predict the flux. To look at the background, we look at these type of studies that was done by Palson, Small, and many others from the early 80s. What they used was they had a set of reactions for the entire cell. Those set of reactions, to solve all those reactions, you have to use a method in mathematical terms called as linear programming. One second. Linear programming is an optimization method in mathematics where when you have a set of linear reactions or a set of equations, you can find optimal solutions. So that entire thing ended up creating this entire field of flux balance analysis. So what do we need for the flux balance analysis? You need to know the genome. You need to predict the metabolic network. If that particular organism doesn't have a metabolic network, then you have to do a metabolic network reconstruction. Then infer which metabolites can be produced, what precursors are needed, then give the number of nutrients, what are the hypothesized requirement, like for example, your biomass production. Then you predict the optimal flux distribution in the network. When you have this, then you can find out what are the major pathways, what are the minor pathways, how the flux travels. If a flux travels in a um, very constricted way in a particular pathway, then probably that pathway or the targets in that particular metabolic pathway are good for drug designing purposes. Okay, so I'll just go to this particular equation. N, we saw it as stoichiometric matrix, and V is the vector of steady state reaction rates. Okay, when you do a dot product of N and V, you should get 0. If you don't get 0, that means your stoichiometric matrix is not balanced, which means this is not in a steady state. So when you look at a flux space, the flux space, what we call is nothing but a multidimensional space, and each dimension is a reaction. And I will just show you example here. Let us say A, B gives rise to X and X gives rise to C. So there are three reactions, four metabolites. If we balance the equations and let us say this is the flux space and the pointer that I'm pointing is the white space here. Now the reaction 1 is in this direction, reaction 2 is in this direction, reaction 3 is in this direction, and all three are pointing in different directions, and whatever space within which these three reactions are taking part, you get this flux space. Now, based on the stoichiometric matrix and the ODEs that you get, what you will get from in this multidimensional space is a polyhedron, which is shown in this blue line. This polyhedron here, it's shown as a rhombus. You will have on the surface of the polyhedron the solutions. Now the solutions will have a minima and a maxima. To find which solution is a maxima and to find which solution is a minima, you need the mathematical operation of the linear programming. And there are many algorithms that are well defined that do the linear programming. So using linear programming, we can see that in this polyhedron, the solution is at this topmost corner where this black dot is present, where I'm placing my pointer. So there is one solution which is present within these constraints. So this particular shape of this polyhedron, it's a rhombus here. We call that as a flux cone. More constraints you add, you will have the solution space reducing because you will have a multidimensional polyhedron. Okay. So depending on the direction of the fitness gradient, you will have the optimal value of the flux either in the corner or in the maxima of the entire surface of this flux cone. Okay. So the steps that are used in flux balance analysis is that first you define the system just like how we did earlier. You take a pathway, you set the limits, you see if it comes in a steady state, and then you define or you change the limits, and then you see if it is in a steady state. Then you do the mass balance, which is by constructing the stoichiometric matrix. Then you define the measurable flux, and then you do the optimization, which is the linear programming. So, in the case of an organism, you need to know the genome. When you take the genome, you need to know what is the coding sequences. When you look at the coding sequences, you will do an annotation. You will see the function of the protein. You will have a lot of hypothetical proteins. You will have a lot of putative proteins. Product of each of the gene is a species or the reactant in the network. Reactions are those that the gene product associations, like for example, this particular genome, let us say there are 10 gene products that take part in glycolysis. That means there will be 10 reactions for which each of those genes can be correlated with or you can associate with. So you have to come up with all these reactions. And for many of the model organisms like E. coli or Bacillus subtilis or Saccharomyces cerevisiae, metabolic network already exists in KEGG, Brenda, and other databases. So you can directly take those reactions, but then it doesn't mean that everything will be present. There will be some reactions which will be missing. So you have to construct that model according to your requirement. So then, sorry, then you have to look at what transport mechanisms are present, which reactions occur through diffusion, which reactions are through the porins or outer membrane proteins. So do you consider them as part of the system or not? All these things have to be answered, and that again depends on the particular pathway. So here you have a metabolic network, then you make the limit, that is the system boundary, which is shown in yellow line. And now once you have the system defined, then you do the mass balance by ODEs and the stoichiometric matrix and the various fluxes and the various uh, vectors. Okay, so you have the stoichiometric matrix and the reaction rate vector. Now, as I said, the S dot V is should be 0 only when you get the dot product of the stoichiometric matrix along with the reaction rate vector here. If you get a value of 0, then you know that you have balanced it and it is at a steady state. Once it is in steady state, then when you start looking at the solution space, this is the solution space in 3 dimension, but actually, depending on the number of reactions, you have multi-dimension, and then you get a flux cone. Now, once you get a flux cone, you are trying to maximize the solution, and that is denoted by an objective function called Z. This objective function is what we solve using the linear programming, and then we find an optimal solution. It's not necessary that you will get an optimal solution in the first go. Sometimes you have to change the system, sometimes you have to change the values, sometimes you have to include different kinetics, you have to look at different literature, come up with a lot of different, solve a lot of different variables and so on. Then sometimes you might have to add additional constraints which may not be defined. So then in that case, like for example, you have rate constants which tell you that this is the space in which the solution can be present. But then you have different other optimization. I'll just show you that figure and I'll come to this. Okay, so for example, these three white lines are the constraints. The solution will lie within this constraint. But sometimes when all constraints are present, the solution may not be possible within all the stated constraints. And that depends on the system. Okay, and in some cases, you will have to add other types of data like transcriptome data, proteome data, metabolome data. The more you add, your solution space will shrink. So you can see the color coordinating with the data that's added. The large one is the genome data. Then when you add the transcriptome data, then your solution space is this much. Your proteome data, when you add the solution space is narrowed down further. When you add metabolome, it is further here. Now, within this green area is where you have your constrained solution space, and the maxima is present here where there are biologically relevant solutions. Now imagine if you are putting too many constraints and the solution is way beyond the constraints. Your solution lies outside these constraints, then you'll have to remove some of these constraints. So the solution is only as good as the constraints used. So it's not necessarily the more the constraints you get a good solution. It depends on the system, depends on the question that you're asking. So you can use mutant studies, you can use protein truncation data, some other biochemical data using which you can come up with constraints. So from the model, we balance it, we get the activity of the vectors, then you compute the steady state, then you add the constraints, and then you get the optimal solution. So these are the steps in which the flux balance analysis is done. Okay. I'll skip all this. Okay, so now I'm going to show you a case study how in Mycobacterium tuberculosis, how they went ahead and identified the drug target using systems biology approach. Okay, before I go, any questions up to this point?
Sir, these ODS and stoichiometric analysis is for metabolic pathways only, or is it related to all multi-omics?
Omics is genomics, proteomics, transcriptomics, metabolomics. Okay. So when you're looking at an organism's regulatory network or organism's metabolic network, then when it is in a network, you can define it in chemical terms like something is getting converted to something. Then you can construct a ODE.
Okay.
Okay, so just with the omics data, you cannot construct a ODE because you need to know which particular entities are taking part in a reaction, right? So until unless that reaction is understood or the relationship between the entities is understood, you cannot construct a ODE. And just because two entities or two genes are related or two genes are taking part in a reaction doesn't mean you can construct a ODE and solve the reaction, because there must have been some rate constant also available. If the rate constant is not there from literature or from empirical data, then you have to hypothesize that they are probably taking part in a reaction.
Okay, so this might be applicable in any of the rate dependent reactions only, sir?
Yeah, you can apply the construction of the model. Whatever I talked earlier is computational model, and that is one way of constructing a computational model to understand the systems or the system of the particular organism. As I said, you have partial differential equations, you have quadratic equations, you have Boolean network or you have Boolean model, you have network perturbation analysis. There are many different ways in which you can understand a system and you can get more insights. This is one way with which where we look at substrate product kind of relationship and come up with a computational model.
Okay, so using this, can we identify any disease-specific gene expression and also difference between housekeeping gene expression?
No. Those things are experimental. You
Okay.
cannot find expression values through this. We are not looking at those expression values here.
Okay.
When we say one substrate with another substrate gives two product, we are not saying one protein plus another protein gives two protein. That 1 and 2 is just a coefficient. It doesn't mean there's only one entity of the protein in the cell. That might be more than one entity of protein in the cell. The number of protein or number of copies of mRNA and protein is completely stochastic. That changes from cell to cell.
Okay.
Any other question? Okay, so in this Mycobacterium tuberculosis, what they did was that they took this mycolic acid pathway and then they constructed a flux balance analysis study, and they looked at what all can be potential targets for anti-TB drugs. Okay, so if you have to design a anti-TB drug, then what targets can be targeted? So here in this Mycobacterium tuberculosis, in this particular model, they took 197 metabolites that participates in 219 reaction, which is catalyzed by 28 proteins. This is not the entire Mycobacterium tuberculosis genome reconstruction. It is just one part of the organism's network that is taken up by 28 proteins. So this is the mycolic acid pathway. So it undergoes, it has many different components, many different systems. 1 second. Okay, so there is a lot of different, sorry, it's the same image that has been overlapped on this, but there is many different parts. They have identified this entire network into many different sections. So each is kind of independent, but then they are connected with each other. So when they looked at this particular model, they deleted some of these genes in the network. When they deleted some of these genes, they saw how the flux travels. As I said, remember the example, the national highway, if it is blocked, you have to go to Delhi, but you take a alternate route. You see if by through a neighboring village you can reach or you can get over the highway, the traffic jam. So when this without gene deletions, this is the relative flux. Okay. Now, when this particular gene was deleted or when this particular gene was deleted in the model, this is again computational. You don't have that particular gene present in the equation. Then what happens? You see that the flux is completely non-existent, but here the flux still exists very close to your wild type. In 90% inhibition of this particular gene, you do not have very similar expression as the complete deletion. So that tells us this particular gene, if you delete, you are actually creating a good drug target, or this drug target is validated by the presence uh, of which particular gene is essential, which particular gene is non-essential. So using this 28 proteins, the list, you can find out what the essential genes are, what are the non-essential genes are in this particular pathway. And that's what they did. So using this particular, all this flux balance analysis was reported in the PLOS computational biology paper about 20 years ago. Then they also did the pipeline for adding other omics data, intractome, reactome, and genome level of the same in the TB, Mycobacterium tuberculosis. So this was done by Karthik Raman and Nagasuma Chandra's lab when he was doing his PhD. So this is the pipeline they had come up with. So you have the entire genome of the organism. You take the network of the metabolites, you do the flux balance analysis, you find out which genes are essential. You can do single gene deletion, you can do two gene deletions, you can do three gene deletions, you can do multiple gene deletion modeling, and then you can also incorporate structural data that using which you can find out whether the expression level of those genes is changing in different conditions. What is the similarity of those targets that got finely filtered from all these steps, whether they are present to they are similar to other targets with in terms of structure, whether they are similar to other proteins present in the gut organism. Because if you're designing a drug against tuberculosis, that same drug can have non-specific binding to other proteins with other organisms. So using this, they found out what were the unique tuberculosis targets that are present mainly in mycobacteria, and what are the broad spectrum targets that are present in other pathogens. So you can have a broad spectrum drug or a narrow spectrum drug that can be identified. Okay, so this is again, they identified Mycobacterium tuberculosis targets, specific targets, broad spectrum targets, targets that are implicated in persistence. Now, why persistence? Because Mycobacterium tuberculosis is a bacteria that can form biofilm, and it can remain in the body, in the human body, for long periods of time, and through a mechanism of biofilm persistence. Okay, so using a system level analysis, sequence, genome, and comparison with structure and other proteome data, they came up with this. So this is the final list where they've said that, okay, these are the gene targets that are regulatory in nature, lipid metabolism, cell wall. All these are in a way drug targets, but they are all Mycobacterium tuberculosis specific. Some are strain specific. Whatever you see within the parentheses are specific to that particular Mycobacterium tuberculosis strain. So and so on. You can find out what are the drug targets. So you can replicate this for any organism, any pathogen, and you can find out whether that particular pathogen's drug target is identifiable. Does it have any systems level identity? And you can also find out what the pathogenicity of the target is, and so on. Okay, so target identification using systems biology approach is a routinely used method, and it has been shown with Mycobacterium tuberculosis, and there are many other different pathogens for which the systems biology approaches have been used. With that, I'll stop. If you have any questions, you can ask.